Today we are going to perform the experiment to study the plant density by quadrant method. This will perform under the lab condition, that is inside the laboratory or the classroom. For this we require a cardboard. As you can see, this cardboard is divided into the 4 quadrants, which are marked as quadrant 1, 2, 3 and 4. We will take 4 different types of pulses which represent the 4 different types of plant species. Let the A plant species represented by the white chana, and B by this groundnut, C species represented by this black chana, and D by this moong dal. Right? So you can take any type of pulses, any 4 type of pulses which represent the 4 different types of plant species. Now we will scatter these all pulses on this cardboard, that's a quadrant, and then count the number of each pulse in each type of quadrant. This is how we have spread. Now we will count each type of the pulse in different type of quadrant. So the pulse which is lying at the boundary, we will exclude that. It should be inside that particular quadrant. So if you will see, let's count the species represented by the white pulse. See here, this is your 1, 2, 3, 4, this is 5 and 6. So this will be 6. Likewise in quadrant 2, let's count it. So there are 3, 4, 5. Okay. In third is 3. In fourth there are 2. We will maintain a table of population density to record our observation. The first you can see serial number, the name of the species, the number of individual in the quadrant, each quadrant. So 1, 2, 3, 4 quadrant we have written. Then total number of individual in a quadrant, and in the last column we write down or calculate population density. The formula for population density is total number of individual of the species in all quadrant divided by the number of quadrant we have studied. This way we will note down in a tabular form the number of species A plant present in each quadrant. Just we have seen species A plant in quadrant 1 are 6, then 5 in 2, and 3 in 3, and 2 in 4. Likewise, you will count the number of B species C and D in each quadrant and note down in this table. Yes, so you will count each and every type of plant species representation over here in different type of quadrant, and then complete the given table. Now, so this is the number of species. For A plant we are taking in quadrant 1 we are getting 6, just we have counted. In second 5, and third में 3, and fourth में 2. Now here we will total this one. So this will be how much? 6 plus 5 is your 11. Then 14, 16. So 16 is this is 16 total. Now we will calculate the population density by applying the total number of species in all the quadrant, that is your 16, divided by number of quadrant we are taking, total 4. So this will be how much? 4. So this population density for your species A is 4. Likewise, we will calculate or count the B species in every quadrant, every 4 quadrant, write it total and apply the formula. This we do as it is for all the 4. Then we compare the population density for these 4 species. So in this way, our observation table is ready, and we have calculated the population density for different species, the 4 given plant species. Now we will compare it. See, it is 4 for species A, 1.5 for species B, for C it is 10.5, and for D it is 9. So So it is clear that in the field, that in the 4 quadrant, the 4 species are present. The average population density of plant of species C is higher and plant of species B is lowest. Therefore, species C is the dominant and other species studied in the community. So we have to report in the end which species is the dominant and which is not. The species C is dominant on other species studied in the community.